Hey everyone, and welcome to This Explainer. Today, we're taking a journey that literally shrinks us down to the microscopic level. We aren't just going to be looking at some static, boring textbook blob today. Actually, scratch that. We're touring the cell as a bustling, highly organized, and relentless microscopic factory. So get ready to see the fundamental building blocks of life in a whole new light. Here's our quick tour map for today. First up, we'll go from organisms to cells. Then we'll head straight inside the cell factory. Compare prokaryotic versus eukaryotic models. And finally, contrast plant and animal cells. Let's get right to it.
But wait, before we shrink down, you might be wondering, how do tiny cells perform so many activities? I mean, they're incredibly small, often 100 times smaller than a single grain of sand. Yet they produce energy, they synthesize materials, they divide, and they communicate. How does something so tiny act as the fundamental unit of structure and function for literally all living things? Well, let's find out.
All right, part 1, from organisms to cells. Biology basically operates on a strict corporate ladder. Down at the very foundation, we have the cell. In complex, multicellular organisms like us, millions of similar cells coordinate to form tissues. Then, different tissues organize into specialized organs. And finally, several organs sync up into organ systems. It's like how your nasal pores, your trachea, and your lungs all work together to form your entire respiratory system. But no matter how complex the organism gets, remember this: the cell remains the ultimate boss. Moving right along to part 2, inside the cell factory. We're bypassing the organ systems entirely and heading straight onto the factory floor. Just like any efficient manufacturing plant out there in the real world, a cell relies on highly specialized departments to stay alive. We've got divisions dedicated to building materials, managing waste, and providing power. So, let's meet the key players. First up, we have the nucleus. You can think of this as the CEO's office. It's heavily fortified by a double-layered membrane, using specific pores basically like security gates to carefully control who comes in and out. Most importantly, it houses the DNA. That's the master blueprint and coded instructions for the entire factory. Literally every single activity that happens on the factory floor is guided by the instructions kept safe right in here. So the CEO sends out those genetic blueprints, right? But someone has to actually build the product. Enter the ribosomes. Scattered freely in the cytoplasm or docked on other structures, these are the literal manufacturing machines of the cell. They read that genetic code and synthesize the proteins that actually keep the organism functioning. They're tiny, but absolutely crucial. Now, every factory needs an assembly line, and here that's the endoplasmic reticulum, or ER for short. It's this massive, sprawling network with two distinct divisions. The rough ER is studded with those ribosomes we just talked about, so it's focused entirely on protein synthesis and secretion. Meanwhile, the smooth ER, which doesn't have ribosomes, handles the synthesis and storage of fats, lipids, and certain hormones. Okay, so once proteins and lipids roll off that ER assembly line, they hit the factory's shipping center, the Golgi apparatus. Built from these cool stacks of flattened sacks, the Golgi chemically modifies, sorts, and packages these raw materials into tiny transport vesicles. Whether the product is staying inside the cell or being shipped way out, the Golgi ensures every single cellular package has the exact right molecular shipping label. But here's a thought. Any massive manufacturing process generates dangerous byproducts, right? With all this building and packaging going on, how does the cell prevent toxic waste and broken down machinery from piling up and poisoning the whole operation? Well, the answer is the lysosome. This is the factory's dedicated waste management and recycling center. They are membrane-bound sacs filled with incredibly powerful digestive enzymes. To put that into perspective, if a lysosome ruptures, those enzymes can actually digest the entire cell from the inside out. Kind of scary, but when contained, they safely break down unwanted proteins and worn-out organelles. When you put all these departments together in a typical plant cell, it's quite the sight. You'll spot the nucleus, that extensive ER network, and the Golgi all working in tandem. But the standout feature? That has to be the massive central vacuole. In plant cells, this giant vacuole is the primary storage unit for water, and it generates the internal pressure that keeps the entire plant crisp and upright. Now, if we pivot to a typical animal cell, the layout shifts a bit. It relies on the exact same complex, membrane-bound departments for energy and manufacturing, the nucleus, the mitochondria, the the Golgi. However, it noticeably lacks that massive central water tower we just saw in the plant cell. And as we're going to see in a second, it trades a rigid outer wall for a far more flexible border.
Which brings us to part 3, prokaryotic versus eukaryotic models.
So we've got two main franchise models here, and the differences are absolutely fundamental. Eukaryotic cells, the ones building plants and animals, are the advanced megafactories. They range from 10 to 100 micrometers and boast a true, well-defined nucleus and all those complex membrane-bound organelles. Prokaryotes, like bacteria, on the other hand, they are the much older microscopic startups. Pro means primitive and karyon means nucleus. At just 1 to 10 micrometers, they operate entirely without a defined nucleus or any membrane-bound organelles. In one of these simpler prokaryotic bacterial cells, there is literally no protected CEO's office. The genetic material is just a single circular DNA molecule floating openly in a region called the nucleoid. Without specialized rooms or departments, all cellular activities just happen freely in the open-plan cytoplasm. It's incredibly basic, sure, but biologically speaking, it is highly, highly successful.
And finally, part 4, plant versus animal cells.
If you were to prep a slide in a lab, the architectural differences are super obvious right away. Plant cells, like what you'd see in an onion peel or a Rio leaf, pack neatly together, like rigid, highly organized bricks. Conversely, human cheek cells, which are animal cells, sprawl out in a really irregular, kind of chaotic arrangement. So why the difference in blueprints? The reason for this structural difference, it's all in the boundaries. Every cell has a selectively permeable cell membrane. But plant cells wrap that membrane in an additional, rigid cell wall made primarily of a tough carbohydrate called cellulose. This provides immense structural support, keeping the plant upright even if it loses water. Animal cells, though, rely exclusively on the flexible cell membrane. And that microscopic flexibility is exactly what allows animal tissues to stretch, bend, and support our ability to walk, run, and just move around. So to wrap up our tour today, I want to leave you with a quick thought experiment. What consequences would you predict if a plant cell's wall suddenly became as flexible as an animal's cell membrane? Think about the towering trees outside or the potted plants sitting on your windowsill right now. How absolutely vital is that microscopic rigidity to their survival, and what would happen if that factory wall just collapsed? Keep exploring, keep wondering, and thanks so much for joining me on this tour of the cell.